Hey everyone, this is Miss Valentine, and you are watching the Scientific Method Notes presentation. And we are going to take a look real quick at the handout that you're filling out. Today it is a Cornell style notes. So you'll notice there's questions on the left, and then on the right is where you're going to actually write your notes. You don't have to answer the questions under here. The point of Cornell Notes is that you have questions all on one side and then the answers are on the other side. So you guys will just be filling in on the right side here, and then at the end you will have a 5-sentence summary. So let's go ahead and get started. The first part of these notes, I'd like to differentiate between a theory and a hypothesis. So many of us might say we have a theory about something when really we just have a hypothesis about it. We have a guess, an educated guess. It's educated because supposedly with a hypothesis, you've done some research first to see if there's any other information out there. So the hypothesis is an educated guess, and it can become a theory if it is testable and if it has been tested many times. So a hypothesis is more or less a statement of what you think might happen, a prediction, if you will. And then if it gets tested many times and evidence supports that hypothesis, then it can become a theory. A theory has undergone extensive testing by various scientists. So it's important that any scientific data is tested by many scientists, not just one person, because one person could end up being biased. A theory is a generally accepted, accurate explanation of a certain observation or a prediction. So it's generally accepted, but it doesn't mean that it is correct. It means that it can still be questioned. However, a lot of evidence is there to support it. So if it's highly supported, it will be taken as fact, and the theory will be tested continuously, though, to make sure that it is correct. And new information might come to light that could change the theory along the way. In other words, it has to be tested many times so that you can trust the results. And then it is a proposed explanation for a lot of natural things that we see. It is not proven, although you do hear that word a lot in science. Nothing really is ever totally proven. Science is subject to change, and that's one of the cool things about science. A lot of times people, when they're studying science, think it's like this book, this concrete block of knowledge that is unchangeable. But the cool thing about science is that it is actually very changeable and new information comes out all the time. So I just don't want you guys using the word proven because it does indicate that something is set in stone when really it is not. So theories, no matter how well accepted, are always subject to change as new information comes to light. And this happens all the time. So it's not always obvious when you're in high school because a lot of what we teach you in high school is knowledge that has been tested many times. But if you ever go into a career in scientific research, you actually will discover new things, will discover new knowledge, and new theories will come along. All right, so here we are at our first pit stop. So on your piece of paper, you're going to write a complete sentence, and I gave you a sentence starter here. A hypothesis is, so explain what that is, while a theory is, and explain what that is. Answer the pit stop while you pause the video. Scientific models are something that describes or represents nature, and you use scientific models to make predictions. A common model that you often hear about is a weather model or a climate model. So it's something that is meant to represent or show nature, something in nature, even though it isn't actually that thing. Usually scientific models are like a scaled down version of something that is in nature because the earth is huge. The earth is really big and there's all sorts of complex things going on. The model is the best way that we can possibly study certain aspects of science. Models can be changed and refined. In fact, they are all the time. Weather models, like let's say there's a storm coming in Columbus, a snowstorm, and you're hoping for a snow day, they will predict it, and then sometimes it doesn't happen. That's because their weather models may not be accurate. Weather forecasters use, I think in Columbus, it's like 17 different models that they look at, and then they predict based on those models. So models can be changed. They're fluid. They are not permanent, or they can be modified. So all models have limitations, like I mentioned, the whole snow day example. This happened actually probably 5 years ago. There was supposed to be a huge storm coming to Columbus. We all thought we were going to have a snow day, and then we woke up, no snow day. So every model has its limits, like I just said in the previous slide. All right. So we'll move on here to the scientific method. Now I want to say that the scientific method is just one way of doing science, and it's usually used for controlled experiments. A controlled experiment is where you have fixed variables, and we'll talk about those different variables. But there's other types of processes that are used in science. The scientific method works really well in a closed lab environment where you can control things. But it gets more difficult when you are in, say, environmental science, when you're studying ecosystems that don't really have clear-cut edges. You really have to create models to try and run your experiments to test things on. So I just want to be clear, the scientific method isn't the only way of doing science, but it's the best way that we have to do controlled experiments. So what is the scientific method? Like I said, it's one of many processes used by scientists to solve problems. A scientist will have a question they are trying to answer, or in the case of like the COVID pandemic, they're trying to find out what medicine works best to help either prevent COVID from happening in the case of vaccines, or to treat people who already have it. So this is a question, something that they are looking at. And so in these trials, these drug trials that you hear about on the news, they are using the scientific method as a way to solve this problem, to answer those questions. All scientific understanding is based on observing, so using our senses: sight, hearing, taste, touch, and smell. Now, when we do our nature journaling, we're using 4 of those 5 senses, possibly. I don't like you guys tasting the bark on the tree or the grass in the yard, so we'll stick with sight, hearing, touch, and smell. Oftentimes, you can also take measurements using extensions of the 5 senses. So you can use different things like measuring tape, rulers, a thermometer. So those are things that are extensions of the 5 senses that allow you to take measurements that isn't directly from your body. All right, so step 1 of the scientific method is to ask a question or to state a problem. So what is the problem? What are you trying to find out? So I'm going to propose one as an example. We're trying to figure out if caffeine will help you perform better on a test. So that is my question. Next, you want to do some research. Find out what there is to know on the subject. So in this case, what does caffeine do to the brain? How does the caffeine impact test results? What are there other studies out there that you can look at? Your third step is to make a hypothesis. A hypothesis, again, is an educated guess, and the reason why it is educated is because you've done some research. So with your educated guess, you are trying to determine what you think may happen. Now, you aren't like emotionally attached to your hypothesis. It doesn't really matter if it's right or wrong. You're more or less taking a stance, saying this is what will happen. And you can be wrong. It's totally okay to be wrong. A lot of scientists do kind of get like invested in their hypotheses, and that's where we see scientific bias coming in. Take my daughter's home. All right, so the hypothesis is step 3. Step 4 is designing your experiment to test the hypothesis. And this is where we'll get into a little bit more details about what an experiment, a controlled experiment, looks like. So there's very specific variables, a very specific way to set it up so that you can trust the experiment. The 2 types of variables are the independent variable. The independent variable is what is being changed or tested in the experiment. I like to think of a memory trick. The independent person is someone who changes a lot and tests things out. So an independent variable is changed or tested. The dependent variable isn't like an actual object, it's really what you're measuring. And the dependent variable is going to respond to whatever change it is you're making. It's going to respond to the independent variable. So we'll do this as an example. Um, the experiment must include only one independent variable. The reason is you want to make sure that whatever result that you're getting is a result of only that one change. If you change more than one thing, then you won't know which one of them actually caused the result. So the dependent variable depends on the independent variable. The measurements that you get, the whatever it is, temperature, heart rate, you name it, that depends on whatever change it was that you made, increasing your caffeine or turning the thermostat up. I don't know. But the dependent variable depends on the independent variable. So here's our example again. You're measuring the effects of caffeine on, okay, we'll do heart rate instead of test results. So you're measuring the effects of caffeine on heart rate. You give one group caffeine and another group water. So half of the class gets caffeine, half of the class gets water. The heart rate, whatever you're measuring, depends on whether or not you had the caffeine. And the caffeine, again, is what is being changed. One group of students gets caffeine, the other group doesn't. The caffeine is the independent variable. It's what's changing from one group of students to the other. And then the heart rate is what you're measuring. You're checking to see if their heart rate goes up or down. So you're measuring that. That is your dependent variable. So that heart rate is going to depend on whether or not you have caffeine. The experiment must also include a control group, and this is why it's called a controlled experiment. A control group is set up exactly like the setup with the variable, with the changed thing. In this case, caffeine is what we're testing. But it does not contain that thing. It doesn't contain that change, that caffeine. So controls are factors that stay the same throughout the experiment. You keep everything constant, you just change that one thing. And the control group is the one that does not have the change. In the above example, in the example about the caffeine and how it affects your heart rate, there is a control group, and it is the group without the caffeine. To create a control group, all you need to do is take away the independent variable. So you take away the caffeine, the thing that you are changing. When you, I believe on your test, there's a question about how many setups you need for an experiment. You must have, for a controlled experiment, you must have at least 2 setups. You need at least 1 setup with the independent variable and 1 setup without the independent variable. So there's a minimum of 2 setups. You can always have more, like you could test different amounts of caffeine if you want to, but you always have to have that control group without the caffeine and the group. It's oftentimes called the manipulated group or the experimental group that has the caffeine, that has the variable. All right, so here is our next pit stop. You can pause the video to answer. What is the difference between the independent variable and the dependent variable of an experiment? And make sure you do complete sentences on your notes paper. After you've done your experiment, or I guess this is more or less during the experiment, you are going to observe what you see and collect data. You can record data using your visual observations, just what you see. You can take pictures of what you're seeing. You can take other measurements, like specifically using a measuring tape or other types of instruments. You can create graphs, data tables. So all of that is involved in the experimental process. You design your experiment, and this is the actual running of the experiment, making your measurements. And I'll leave this screen up for just a moment since I know you'll probably be copying it. After you've collected all of your data, the last step, second to last step, is to take a look at that data and determine what happened, and then try to explain why it happened. You can also ask questions, okay, does this data look kind of funny? Is this reliable? Is this credible? So the analysis step is taking that data that you have and looking at it and seeing what you see. Oftentimes, you'll be analyzing some of the graphs or the tables to see if there are any patterns. And then if there's something funny that just doesn't seem right, you might talk about that in your analysis of the data. The final step is the conclusion, and it's not like a conclusion in English class where you say, in conclusion, da 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 da. A conclusion is just stating whether or not your hypothesis was supported or rejected. And again, it doesn't matter if it gets rejected. A hypothesis is just taking a stance on something, and then after you've collected all the evidence, all the data in your experiment, you just say whether or not the evidence supported your hypothesis or your stance, or if it rejected your hypothesis. And from there, you can go on to create a new hypothesis and start the process all over again. And that is it for our scientific method notes. So you guys are going to fill in the summary section at the very bottom with 5 complete sentences. When you're finished, you can hit the submit button if you're doing an electronic version, or you can turn in the notes after you take your test this week for chapter 1 and 2.